What do diseases such as schizophrenia and depression, and the actions of therapeutic or recreational drug use, have in common with fundamental neurobiological processes such as experience and learning? The answer is not as complex as you might think. Although the details are very specific, the underlying biological mechanisms are very similar. Synaptic connections are changing their strength. So how is this happening? Thinking back to what you've learned earlier in the course about synapses, very generally speaking, changes in synaptic strength can be due to either A, structural, or B, non-structural alterations, the synapse connecting the presynaptic and postsynaptic neuron. Structural changes of synaptic strength, which for example include the alteration of number by loss or addition, or size by growth or shrinkage of synapses. These occur normally throughout life, but are of particular importance during development, when the brain grows massively in number and size of neurons. And synaptic connections between these neurons are being shaped and reshaped constantly. But development isn't the only stage in life where processes like this are important. In addition, structural changes occur at the end of life as a part of normal aging, and when things go wrong, as in the case of certain neurodegenerative disorders like Alzheimer's or Parkinson's disease, where neurons either lose their synaptic connections or die off completely. The vast majority of these changes in synaptic strength during the human lifespan, however, are not structural in origin. Rather, they're induced by changing synaptic strength. The scientific term for that is efficacy, without affecting the structure of the synapses involved. Instead, these changes in synaptic strength are mediated by a broadly defined mechanism called neuromodulation. So what exactly is neuromodulation, and how does this process work? Generally speaking, neuromodulation is a process not unlike classic synaptic transmission of chemical synapse. However, instead of only two neurons being involved in information exchange, we now have to consider the action of three or more neurons. So what makes neuromodulation different? It's critical to understand that in a modulated synapse, nothing changes structurally in the unidirectional flow of information from the presynaptic to postsynaptic neuron in the way we've learned for synaptic transmission. Instead, the only property that's being changed is the strength or efficacy of the synaptic connection through an increase or decrease in the size of the postsynaptic potential due to the action of a third neuron. This change in synaptic strength can have its origin presynaptically, for example by altering the number of neurotransmitters released, or postsynaptically by altering the response of the postsynaptic cell to the neurotransmitters. But as you already know, there's nothing in neurobiology that's black and white. Both presynaptic and postsynaptic mechanisms can of course work together and result in altered synaptic efficacy between two neurons.